Hello, everybody, and welcome back to BioSC 140 Human Physiology. This video is going to cover the discussion part of Laboratory 22, red blood cells and blood typing discussion. All right, so let's get right into it. Mature red blood cells lack a nucleus. So we're going to talk a little bit about some background information on red blood cells to start out. So mature red blood cells lack a nucleus. Now, if you've been following along in the lecture, you should know where red blood cells come from. When students first hear this, a lot of them think, no nucleus. How do we get more red blood cells if they have no nucleus? Well, the answer is they come from pluripotent hematopoietic stem cells. And when those pluripotent hematopoietic stem cells get the right signals, the right stimulation, they start down a maturation process, which leads to the production of erythrocytes or RBCs. Now, remember, I think it's really important that you speak physiology, that you speak healthcare. And when there are multiple common names for something, I want you to know all the names. You will see all the different names mixed in the exams. So erythrocyte is an RBC or a red blood cell. They have a characteristic shape. Most people, when they see a red blood cell, they go, that's a red blood cell, because they've seen it in movies, they've seen it around. It's got this classic biconcave shape. Now, the benefits of this, form and function, going back to our first day of class, are that it increases surface area. Now, that also goes back to our first exam, our first section. What law said that when you increase surface area, you increase diffusion or increase flux? Fick's law. As we increase the surface area on the red blood cell, it speeds up the diffusion of gases in and out of the cell. Red blood cells are flexible. Now, in a couple labs, we're going to actually see video of red blood cells flowing through arteries, veins, capillaries, and you're going to see them bouncing off each other. You're going to see them speeding through the tunnel and hitting the sides. You're going to see them squeezing through capillaries. They're flexible because they got to squeeze through capillaries, and they're always bouncing back and forth off each other. In that lab in class, we actually take a live goldfish, we put it to sleep, and we put the tail underneath a microscope, and we look at the blood flowing through the vessels in the tail live, and it's incredible. So I'll find some videos of that for you for that lab. And don't worry, no goldfish are harmed. I have yet to not wake the goldfish back up. All right, so red blood cells are anucleate. They don't have a nucleus or organelles. This maximizes space for hemoglobin. Remember, oxygen is not very soluble in water, and our body's way of increasing the carrying capacity of our blood for oxygen, its response are red blood cells and hemoglobin. Hemoglobin or red blood cells are basically sacs of hemoglobin. During that process of maturation from pluripotent hematopoietic stem cell to red blood cell, the reticulocyte is the name given to a maturing red blood cell. So it's immature at this time, but it's in the process of maturing red blood cell with a nucleus. I do want you to know this. There are clinical applications. Functions carrying O2 and CO2, carrying gases. So we used this hemocytometer blood cell counter to figure out how many red blood cells were in a unit of our blood. Well, what did we do? We took a short capillary tube, we filled it up completely with blood, no air, because we need to know exactly how much blood was in there, so that we could do some math, we could do some dilution factors. We then put that blood into a vial containing a known or a little cylinder knowing with a known amount of liquid within the cylinder. And that liquid contained a substance that lysed every cell except for red blood cells. We then took a micropipette and we added some of that liquid with red blood cells with blood onto this hemocytometer. We dropped it right into these little V-shaped things on the sides of the hemocytometer. And then we looked at this graph, this grid pattern underneath the microscope, which is right here in the middle of this hemocytometer. And we counted all the red blood cells in this square 1, square 2, square 3, square 4, and square 5. And we multiplied by 10,000 to come up with our total red blood cells. Normal is about 5 million. It's a little bit higher in men usually than in women. This photo right here, that's what each one of these squares will look like when we look at it underneath the microscope in lab. We did hematocrit. So hematocrit is the packed red cell volume. It's when we take a sample of blood, this tube right here, and we spin it in a centrifuge, what percentage of the whole is the packed red cell volume? So when we spin a capillary tube that's full of blood, we get all the red cells, because of centrifugal force, they all get pushed to the outside and get packed down. On top, we have this yellowish plasma, the liquid portion of the blood. What percentage of the whole is the packed red cell volume? So right here, hematocrit is equal to packed red cell volume, down here, divided by total blood volume, 100%. Given as hematocrit or percent plasma, and can calculate the others. So plasma plus hematocrit should equal 100%. Normal is around 42%, but it varies. It could vary because of hydration levels. A number of different things can cause it to change slightly, but it should be above 37. It should be above 37 or above. Anything less than 37 is considered anemia. When we have our spun tubes, there's a few different methods that we use to figure out the hematocrit. You could pull out a ruler and do some math, or there are a few tools like this one right here, which will tell you the percentage pretty easily. We have another one in lab that's kind of like a sliding graph. There's a photo of it in the worksheet, and once you see it, it'll make sense. But it's basically just, it makes it so you don't need to use a ruler and do math. It just goes a little bit quicker. All right, so let's talk about hemoglobin. Hemoglobin is the main component of red blood cells. Hemoglobin, HB, is a protein found in red blood cells. There's 280 million hemoglobin molecules in a red blood cell. That's a lot. 280 million per red blood cell. Then we have 5 million about red blood cells per microliter of blood. There's a lot of hemoglobin in our bodies. It's usually around 14 grams per deciliter. Structure, it's a tetramer. So there's 4 subunits within the hemoglobin molecule. There's 2 alpha chains and 2 beta chains. Each subunit has 1 heme group. So 4 total heme groups. Each heme group has an iron molecule. So we need iron to make hemoglobin. And each heme group can bind onto 1 O2 molecule. So we can have a total of 4 O2s per hemoglobin. 1, 2, 3, 4 O2s can bind. Hemoglobin is a carrier protein. Oxygen doesn't dissolve very well in aqueous solution. We need to be able to carry more oxygen than we would if it was just aqueous water, aqueous blood. So we have hemoglobin. to add more oxygen carrying capacity. But oxygen is not the only thing that can bind onto hemoglobin. CO2 can also. And CO2, carbon monoxide, we got the next slide coming up, we'll talk about that. It's also a buffer. So I've asked questions on that before, what are the functions of, or which of the following is not a function of hemoglobin, and so it also has some buffer capacity, hemoglobin does. So right here, hemoglobin carries several different gases. Now, heads up, it's really easy to ask a question about the information on this slide, so make sure you make your flashcards and you're ready for it. So oxyhemoglobin is, the heme is bound to O2, and this imparts a slight red color to it. Deoxyhemoglobin, no O2, has a slight bluish color. Carbaminohemoglobin, it's carried on an amino acid. Carboxyhemoglobin is when you have a carbon monoxide bound to the hemoglobin. So carbon monoxide, CO, is a competitive inhibitor of O2. It competitively inhibits O2. Carbon monoxide basically binds onto these heme groups and doesn't want to let go. It holds on really, really, really tightly. In fact, 200 times stronger than oxygen does. And it can be dangerous. It can kill you. Carbon monoxide, we have carbon monoxide detectors in our homes, because if carbon monoxide is leaked into our homes in a sufficient amount, carbon monoxide has no smell, it has no taste, there's no color. Your bodies have no way of detecting carbon monoxide until it's too late. Or not even then, your bodies just can't detect it. So here in the US, most homes should and do have carbon monoxide detectors. It's kind of like a smoke detector looking device, and some of them are even combined, like smoke and carbon monoxide detectors. Combustion produces CO2. So when you burn wood, there's some CO produced. If you smoke, there's some carbon monoxide produced. So don't smoke. Or talk to your doctor about smoking. Don't listen to me. All right, hemoglobin concentration. So in lab, we use the Tauquist method, to measure hemoglobin concentrations. The Tauquist method, we use a piece of testing paper, and we put a drop of blood on that testing paper. And that drop of blood will change colors, and we compare that color change to this sliding scale right here, this known color in hemoglobin concentration chart. And it'll tell us about how much hemoglobin we have in our blood. Hemoglobin concentration is about 14 grams per deciliter. Men typically slightly higher than for women. Less than 12 is considered anemia. Less than 12 is considered anemia. If you're able to calculate hematocrit, you can guesstimate hemoglobin concentration. And if you know hemoglobin concentration, you can guesstimate hematocrit, because typically hematocrit is 3 times higher than hemoglobin. If you know one, you can estimate the other. So mean corpuscular volume, MCV. I want you to know MCV. I want you to know mean corpuscular volume. This is something that has clinical application. This is something that you might see when you are working in healthcare with a patient in front of you. So mean corpuscular volume, it's the average cell size. How big is the cell? So the way I kind of remember this is like a corpse, like a body. Also, if you speak Romance language in Spanish, cuerpo is body. So it's like the average body size volume of the red blood cell. So MCV is equal to the total total red blood cell volume divided by the number of red blood cells in millions. So if there's 5 million red blood cells, you put a 5 down here. So MCV is equal to hematocrit times 10 divided by red blood cell count in millions. So if it's 5 million, you put a 5. If it's 4 million, you put a 4. Sample calculations, 42 is an average hematocrit. 5 million is an average red blood cell count. So 42 times 10 divided by 5 is going to be 84. Normal range, 82 to 92. 84 is right in the normal range. You need to know the next vocab. It's going to be, there's pretty much guarantee it's going to be on the test, and it has clinical application. You're going to encounter this in the real world with high likelihood. You're most likely going to encounter this. So here's some terminology. So cytic refers to the cell. So microcytic would be like small cell. Normocytic, like normal cell. Macrocytic is like big cell. So if your MCV is below 82, it's considered microcytic. If your MCV is 82 to 92, normocytic. If your MCV is above 92, macrocytic. These terms are used commonly to describe red blood cells, describe the size of the average red blood cell. MCHC is a measure of hemoglobin. So remember when we said hemoglobin bound to oxygen has a little bit of a red color? We're going to describe the amount of hemoglobin, the concentration of hemoglobin in red blood cells using color. So MCV, mean corpuscular hemoglobin concentration, the way you calculate it is average amount of hemoglobin. MCHC is equal to hemoglobin concentration divided by red blood cell volume. So hemoglobin times 100 divided by hematocrit. 14 is an average hemoglobin concentration. 42 is an average hematocrit. 14 times 100 divided by 42 is 33.3. Right in normal range. Normal is 32 to 36. If it's below 32, we call it hypochromic. Chromic's like color. So there's not enough hemoglobin. It doesn't have enough of that red color. It's hypochromic. Normal chromic is going to be normal color. Hyperchromic is going to be above 36. Too much hemoglobin. It's too red, too dark. When we describe anemias, we're going to use both of these words. So when it comes to describing anemias and red blood cells, usually anemias are described either, there's kind of two naming classifications. There's a naming classification that tells you why the person has anemia, so like pernicious anemia or iron deficient anemia. Sickle cell anemia tells you what's going on, what's causing it. But when a patient presents, when a patient shows up in your office, they're not going to show up and say, "Hi, I have iron deficient anemia." Well, unless they've already been diagnosed. But if they're undiagnosed, they're not going to show up and just tell you, "Oh, hi, I have pernicious anemia." They're going to show up, they're going to have their signs and symptoms or not, and you're going to do some blood work. And the lab is going to come back and the lab is going to say, your patient has macrocytic hypochromic anemia. And then it's your job as a healthcare professional to figure out what's causing that macrocytic hypochromic anemia. So this, the macrocytic, microcytic, and the hyperchromic, they describe what the cells look like, not what's causing it. So you're going to encounter both those naming styles when it comes to anemias and red blood cells. So oxygen carrying capacity of blood is determined by several factors. The amount of oxygen carried, the capacity, is determined by several factors. One is the number of red blood cells, the hematocrit, and hemoglobin concentration. If any of these are too low, it can lead to anemia, which is characterized by not being able to carry enough oxygen around your blood. So anemia, compromised ability to carry O2, not enough, can't carry enough oxygen in your blood. Indicators: low red blood cell count, so not enough red blood cells; low hemoglobin concentration, so low hemoglobin concentration; or low hematocrit. Causes: reduced number of red blood cells, insufficient hemoglobin, or inadequate hemoglobin. The hemoglobin is not functioning properly. So reduced red blood cell numbers. So we've talked about how red blood cells are created. They come, in lecture at least, they come from pluripotent hematopoietic stem cells. They're produced at a certain rate. Your body will, and that rate can vary, but your body will be producing red blood cells at a certain rate. They live for about 90 or so days, and then they're recycled. They're destroyed. There's a balance normally, where the rate of creation equals the rate of destruction. If that balance gets thrown off, you can have anemia. If you decrease production, but destruction stays the same, it can lead to anemia. So dietary deficiencies in iron, B12, cobalamin, that's pernicious anemia, or folic acid, or bone marrow disease, because that's where the pluripotent hematopoietic stem cells are, or inadequate EPO. EPO is erythropoietin. We talk about this in the blood lecture aspect. EPO is the signal that's sent to those pluripotent hematopoietic stem cells that tells them you should become a red blood cell. Increased destruction or loss, so weakened red blood cells that lyse, that break easily. This could be due to hereditary spherocytosis. This is not a normal looking red blood cell right here. Spherocytosis, sphere, so sphere shaped cell right here. Or increased blood loss, and it really doesn't take a lot of blood loss. It can take a very small amount of blood loss to lead to this. Menstruation can lead, just that amount of blood can lead to anemia, or having ulcers can cause enough blood loss to lead to anemia. Insufficient hemoglobin, so iron deficiency. There's an iron atom in the middle of every heme group. You need 4 heme groups to make a hemoglobin molecule. If you don't have enough iron, either you're not absorbing it well, you don't have enough iron in your diet, or with chronic infection, your body can kind of sequester all of your iron in your liver to try to starve out the infection of iron. It can lead to iron deficient anemia. Inadequate hemoglobin, so not functioning correct hemoglobin, sickle cell anemia. So anemias are characterized by cell size and color. This is what I was talking about earlier, how we name cells as microcytic, hypochromic. We always use 2 words, one to describe the cell size and one to describe the amount of hemoglobin. So what would this cell right here, the one that I'm circling, how would you describe this cell right here using this naming mechanism? Hyperchromic, it's too red, too much hemoglobin. Normocytic, it's just the right size. What about this one right here? How would you describe this cell right here? So it's too small. So microcytic, not enough hemoglobin, hypochromic. And these are described using a combination of RBC size and color. So we find out the size component by calculating mean corpuscular volume. If the mean corpuscular volume is too low, microcytic. If it's just right, normocytic. If it's too high, macrocytic. So mean corpuscular volume is how we figure out the cytic aspect, the size aspect. Mean corpuscular hemoglobin is how we calculate the chromic part. So we have too much hemoglobin, hyperchromic, just enough hemoglobin, normochromic, too little hemoglobin, it's hypochromic. So given an MCV, mean corpuscular volume of 79.8, and an MCHC of 28, how would you describe this cell? Well, 79 is too low, so it's microcytic. And 28 is too low also, so it'd be microcytic hypochromic anemia. Be ready for a question on the test where you have to calculate MCV or calculate MCHV given values that were could have been determined in lab, and then putting these vocab words to that value. Be ready for that. Know the normal ranges for MCV and MCHC. So iron deficiency is the most prevalent cause of anemia, causes microcytic hypochromic anemia. So iron deficiency would be the etiological term to describe it, what's causing it. And then describing the cell size and color would be microcytic hypochromic. It's the most prevalent, 20% of women, 3% of men, 20% of children, 50% of pregnancies. Pernicious anemia, or B12 deficiency, or cobalamin deficiency, causes a macrocytic normochromic anemia. Macrocytic normochromic anemia. If you remember B12, we talked about B12 also in our digestive tract, our gastrointestinal system video near the beginning of the semester. And we talked about how B12, it could be a dietary issue, usually with vegans, because B12 is found mainly in animal products. Vegans either have to supplement, or I think their two main sources of it are nutritional yeast, and I think some seaweeds, and maybe some fermented products, like fermented mushroom products, maybe have some B12. I'm not vegan, I don't know. But it could also be an absorption issue, not just a dietary issue. All right, red blood cells have surface antigens. So antigens is short for an antibody generating substance. So it's an antibody generator. Antigen generates antibodies. So when your body is exposed to an antigen, your body will make antibodies in response. When your body is exposed to antigens or antibody generators, your body will generate antibodies. It's a substance that stimulates an immune response in the form of antibody formations. They're cell surface molecules, proteins, carbohydrates, or glycoproteins that give red blood cells a unique identity. You've probably heard of this system before. The A, your blood type. Are you A blood, B type blood, AB, ABO, A positive, B negative. This is referring to the presence of these proteins and carbohydrates on the surface of cells, of your red blood cells. So A blood type has A antigens on their cell surface. B blood type has B antigens on their cell surface. A B has A B antigens on their red cell surface. O, some people describe O as a lack of them, but O is kind of like the base. So you have like the O antigen, and then you add an A like on top of that antigen to get A. You have the O antigen, and then you add like a B unit on top of it to get the B antigen. So O isn't like a lack of antigen, it's just like the base antigen. So A and B have the O antigen just with a little extra on top. And then there's the RH, the plus or minus. RH, sometimes called D, is the plus or minus. Genes determine what your blood type is. On chromosome 9, there's one location that will code for AB, or O type blood. And on chromosome 1, there's one location that will code for the RH plus or minus. Humans have 2 copies of each gene, one from mom, one from dad. Which version of a gene do you have? So genotype is what does your DNA say? Genotype is what are the versions of these genes within your blood. Phenotype is what antigens do we see? What are the results of the genotype? So a single location determines ABO blood typing. We have this single location on chromosome 9. You have 2, one from dad, one from mom. And you can either have A at one or both, or none, B at one or both, or none, or O at one or both, or none. Locus is location of gene. Allele is a version of a gene. So we have 3 alleles, 3 versions of this gene. Single location, so right here. This is genotype, and this is phenotype, the blood type. If you have an A allele, A is going to be in your blood type, phenotype. So AB, if you have an A is going to show up in your blood type. A is going to show up in your blood type, A is going to show up in your blood type. If you have a B is going to show up in your blood type. If you have O, it's only going to show up in your phenotype when we describe your phenotype if you're OO. If you have 2 O copies, 2 O alleles, you're going to have the O blood type. Now, like I said, O isn't the lack of this marker on your red blood cell. It's just you're already going to be producing. So somebody who has A blood type it's like an O molecule with a little extra bit on top. So your body will kind of already has O just with A on top. All right. So blood type is inherited from your parents. So right here we have an AO father and a BO genotype mother. Dad is going to give 1 copy to each. Mom is going to give 1 copy to each. And you can see the possible outcomes. We could have an AO child, an AB child, a BO child, or an OO child with this genotype for your parents. You've probably heard of Gregor Mendel back in Intro to Biology, and we talked about Punnett squares. These genes, you can use Punnett squares to figure out what the possibilities are for your children's blood type if you know the genotype of the parents. So right here we have an AO and a BO, and we can just do our Punnett square. So A and B, AB, B and O, BO, O and O, O and A. Do Punnett square for it. Perfect. So plasma antibodies dictate red blood cell surface antigens. So a person who has A A blood type. A person who's A blood type is going to have A antigens on their red cell surface. They're also going to have B antibodies within their body. They're not going to have any anti-A antibodies. They're only going to have anti-B. And then they're going to have A antigens. So an antigen generates an immune response. You don't want to generate an immune response against your own blood, right? So your body has mechanisms to prevent that. A antigen, you won't have A antibodies. Now, there are proteins on bacteria that are similar enough to these surface cell proteins so that when your body is exposed to these bacteria, your body is going to produce an antibody against those bacteria. These bacteria are going to generate immune responses for A and B. So your body gets exposed to these bacteria and produces A and B. Now, if your body has A antigens, your body is not going to produce A antibodies. It knows not to because it's going to attack itself, right? So it only produces B. So somebody who has the A antigens is going to already have B antibodies. Somebody who has B blood type is going to have B antigens on their red cells. Their body knows it has mechanisms to not produce B antibodies. Their body is going to be exposed to bacteria, and that bacteria is going to cause them to produce A antibodies. It would produce B antibodies, but their body knows not to produce B antibodies, but it'll produce A antibodies. So somebody with B blood type already has A antibodies within their body. Somebody who has AB blood is going to know not to produce A or B antibodies, even when exposed to those bacteria. Somebody who has O blood, is going to be exposed to those bacteria and going to produce A antibodies and B antibodies. Somebody who's OO is already always going to have those anti-A and anti-B antibodies within their blood. Plasma antibodies do not recognize red blood cell antigens. Perfect. All right, so O blood type, no A or B antigens, anti-A and anti-B antibodies. A blood type, A antigens, anti-B antibodies. B blood type, B antigens, A antibodies. AB antigens, no A or B antibodies. The problem with having antibodies for your own antigens is they're going to cause something called agglutination, which is a clumping together of red blood cells. And when you can kind of see how they cause right here in this image, that these red antibodies and antigens are causing this big clump right here. That big clump can clog small blood vessels. It can block small blood vessels, which leads to health issues, which is not a good thing for maintaining life in homeostasis. So agglutination is what can happen if you put the wrong type of blood into a patient. If you put B blood into somebody who has A blood naturally, they already have B antibodies, and those B antibodies will bind onto the B blood that you infused into them, and they'll clump together. If you put a B blood into somebody who has B blood, the A antibodies will bind onto the A antigens on their cell surface and lead to agglutination. It's best to match up blood types perfectly. So if you have somebody, a patient with A blood type, it's best to put only A blood into their bodies. But you can use other types in some situations. So if you have a person who has A blood type, you can put other A blood into them. You can infuse A blood into them. You could also infuse O blood into them because their B antibody that's in a person with A blood type doesn't have any B antigens to bind onto. For A blood type, you cannot add any blood or infuse any blood that has the B antigen on the red cell surface. For B, same thing, just with A. It already has the A antibody, so you cannot put any A antigen into this person. So no A blood, no AB blood into a person with B blood. A person with AB blood, they don't have any A or B antibodies in their plasma. So you could add A blood, you could add B blood, you could add O blood or AB blood, and there's no antigens to bind on and cause agglutination. With O, they have A and B antibodies. You can only add O. O is the universal donor. O you can actually add to any of the blood types, but for O you can only add other O blood. AB is the universal receiver. AB can receive any of the other types of blood. I think it's really important to understand the root of words, and it helps you remember them and understand their meanings. I looked up where agglutination comes from, and it actually comes from, I don't know if you speak Spanish, but the word for church in Spanish is iglesia. And the word iglesia and agglutination come from the same root word, which means like the coming together, the joining together, the coming together of people. A church is where people come together. Agglutination, they come together and they join together. So I think that's pretty interesting, and it can help you remember what agglutination is. So agglutination and iglesia come from the same root word. So right here, you can see again, just what types of blood you can put into other people. You can infuse into other people. And what happens when you put the wrong type of blood in, you get agglutination and the clogging of capillaries. Not good. All right, red cell destruction, blockage of small vessels. So if you get agglutination, you get red cell destruction and blockage of small vessels, which can reduce oxygen and nutrient supplies to those tissues by those small vessels. So AB is the universal acceptor. You can put any other type of blood into somebody with AB. O is the universal donor. You can put O into any other types of person into blood. It's safest though to match blood types perfectly. If you're able to, you always want to match blood types perfectly, but these rules apply if you need to. Right here is a little graph. O can be given to A or B or AB. A can be given to A or AB. B can be given to B or AB. And then AB can receive from all the others. Safest to match the blood type perfectly. So Rh antigen. So this is the plus or minus. This is the plus or minus, which we'll talk about their blood type. It's the Rh antigen. It was actually discovered, does anyone know what this is right here? I believe this is a photo of a monkey about to be sent to space. But it's here. Why is there a photo of a monkey about to be sent to space in this physiology slideshow? Well, it's because rhesus monkeys are where this Rh antigen was discovered. Rhesus monkeys have the same antigen on the red cell surface. So Rh, rhesus monkey. And, we have it too. So this is the plus or minus. It's one location on our genes. And it's a little bit simpler for Punnett squares, a little bit simpler for figuring out how it's passed on, doing your Punnett squares, because it's not just plus and minus. There's only two alleles. There's not three alleles like with ABO. So right here, we have positive and negative genotype dad, positive, negative genotype mom. You can do your Punnett square and figure out the probability of each. genotype in the children. There's a little bit of a difference when it comes to the antibodies with the Rh positive. So with ABO, we're exposed to bacteria that will generate the antibodies for the blood types that we don't have. With Rh factor, there's no bacteria that generates that same immune response. So our bodies don't produce antibodies to the other phenotype of blood unless we're exposed to it. So Rh positive has harmless anti-Rh antibodies. Rh negative does not have Rh positive antibodies. So Rh negative does not have Rh positive antibodies. Now with ABO, we saw that if you had the A antigen, or if you did not have the A antigen, you had the A antibody. That's not the case because with Rh factor, somebody who's Rh negative will only generate that antibody if they're exposed to blood of something that is Rh positive. And there's really only one situation that really makes this a big issue. And that's when an Rh negative mom has an Rh positive fetus. So the mom during her first pregnancy will have none of these antibodies. And the mom will be Rh negative. If the baby is Rh positive, she'll be exposed to these antigens and she'll produce an immune response. She'll start to produce Rh positive antibodies. And it's not an issue with the first pregnancy. But after that pregnancy, she's still going to have those Rh positive antibodies floating around her body. And if she gets pregnant again and that baby is Rh positive, her body can attack the red blood cells of that baby and cause the hemolytic disease of the newborn. So hemolytic, blood cleaving, blood lysing disease of the newborn, HDN, or erythroblastosis fetalis. Active anti-Rh positive antibodies from the mother can attack the Rh plus fetus and cause jaundice. If you watch the blood video lectures, we talk about jaundice in there. Basically, if you have when you recycle your red blood cells, you can have a buildup of a waste product, which causes jaundice. And babies have little baby livers, and they struggle to do this recycling, this cleanup as is. And if you increase the rate of red cell destruction because of hemolytic disease of the newborn, you have an increased rate of red cell destruction, and it just overloads the liver's ability to kind of process all of that, and it can lead to jaundice. It's like this yellowish color. Luckily, though, we have modern medicine, and it's prevented with an rhogram injection of mother with antibodies that destroy fetal Rh plus cells. So we're able to handle this with modern medicine. Here's the Rh antigen on the cell surface with A plus, A minus, kind of when you got them all combined. And with that, we're going to conclude this lecture. If you have any questions, please email me. Please take note of the worksheets that are posted for blood typing and for red blood cells. And if you have any questions on those, please email me also. Have a great day, everybody, and I'll see you in the next video.